When Watson and Crick first published their paper back in '53 about the structure of DNA, so remember, DNA was already known as a molecule, and there was pretty good evidence that it was already the inheritor, the mechanism of inheritance of information over generations, but no one knew the structure. And when they published the structure, they are the ones that first proposed that G's and C's base pair together and A's and T's base pair together. And they said this in their paper: "It has not escaped our notice that the specific base pairing that we propose immediately suggests a possible copying mechanism for the genetic material." So they're very insightful about the fact that if G's always go with C's and T's always go with A's, then if you pulled the two strands apart, you still have all the information there. So we're going to talk about how the cell actually replicates and polymerizes new strands of DNA. So this is DNA polymerization. We've got two strands. One is going to be called the template strand. Now the original, and I should go back to this illustration here. This is the original piece that we're going to replicate. We're going to copy each of the strands. One strand is in blue, the other is in yellow. These are the reverse complements of each other. Okay, so DNA is almost always double-stranded. When we copy it, we're going to pull those two apart. So when I talk about the template strand, I'm talking about one of the original strands pulled apart, and then the new strand is being made alongside that. So the original is the template, the new is the one that's actually being polymerized. Okay? So here is a template strand. This is the 5' carbon down here. And here's the phosphate group on that. So this is the 5' end of this template strand. The next, we always bond through the 3rd carbon through the next 5th, 3rd 5, 3rd 5. So the end one here, this is the 3' end of this strand. And now this is continuing. We're not showing it in the illustration, but this could be a really long strand, really long piece. Okay? The strand that we're going to be polymerizing is going to be a reverse complement of that. And we're going to polymerize multiple bases together. Now the bases that we're going to use, or the nucleotides we're going to use, is going to be a base on a sugar and 3 phosphates. Okay, you can only put a triphosphate form of the nucleotide together and polymerize it. And we're going to add it right here. So on the new polymerizing strand, I've got the 5' up here. This is already base pairing. I've already got a phosphodiester bond here to the next base. And the next one that we're going to put on the strand is going to be tethered onto the 3rd carbon. Okay, so you're always putting a new nucleotide on the 3rd carbon of the existing one already in the strand. So we say that DNA is polymerized from the 5' to the 3'. Okay, just always remember you're putting a new one on the 3rd carbon. Okay. This doesn't happen spontaneously. Now there's a lot of energy contained in this new nucleotide that we're going to put in. It's got 3 phosphates on it. There's plenty of energy stored there to get the bond to be formed, but it won't spontaneously just happen. You could put a bunch of nucleotides in a solution of water and just leave them there, and they're not going to spontaneously polymerize. Okay? It's a pretty stable molecule, and it needs an enzyme to overcome the activation energy, to actually get that energy and form the new phosphodiester bond here. So the enzymes that do these are called polymerases. You can have a DNA polymerase. A DNA polymerase is only going to recognize deoxyribose nucleotides. Okay, so here on the 2nd carbon, there's no oxygen there. This is a deoxy nucleotide. And a DNA polymerase will only grab those. There are RNA polymerases, and they will only grab ones that just have the OH group there on the 2nd carbon. There are some polymerases that can do both. They will grab DNA or RNA and will sometimes select one or the other depending on what's appropriate. So you can have hybrid forms of that. Okay, so the DNA polymerase, its job is twofold. It has to come in and it has to recognize what this base is, the base on the template strand, and allow the appropriate new nucleotide to come in. So on the template strand, we've got an A base, so it's going to recruit in a thymine, or thymidine, because it's a triphosphate. Thymidine triphosphate, allow it to base pair, and then it forms the new phosphodiester bond. It does this by cleaving off the last 2 phosphates. So I'm going to lose these 2 phosphates, and it's temporarily going to become a monophosphate. But then I'm going to form this bond between the oxygen on the third carbon. So I'm going to lose those two phosphates, and I'll have formed this new phosphodiester bond. The polymerase then shifts down one base, reads the next one, recruits in another triphosphate, and you do it all over again. Okay? Yeah?
Did you say if that was polymerase 1 or 3?
I haven't told you which one it is yet. Yeah, any polymerase is going to do this mechanism. Okay, so it doesn't matter. In the context of the chromosome and what's the appropriate one to use, we'll talk about that in a minute. But any polymerase is going to do that. Whether it's DNA polymerase, RNA polymerase, one of the dual workings. The chemistry is always the same. So.
Sorry to ask you this. Could you sum up what you just said one more time? A lot of information.
Yeah. So a polymerase is going to recognize the empty base on the template strand. It's going to recruit in a new nucleotide triphosphate that's a complementary base. And then it's going to catalyze the chemical reaction of the phosphate group with the third carbon oxygen. That's what we mean by polymerization. We've added a new nucleotide on there. Yeah?
Where do the other two phosphates go?
They float off into solution, and another enzyme will grab those and reform a triphosphate if necessary. Yeah. So this is called pyrophosphate, two phosphates together. That's a really common thing in cells floating around. And it's going to use energy from metabolism, like your cells breaking down glucose or breaking down fats and using that energy to form more triphosphates, right? Because if I went in and actually cut that nucleotide out, right, if I actually chewed away at that and removed that nucleotide, I've lost the energy there. So there's another enzyme that has to recharge the nucleotide monophosphates. And so it would grab the pyrophosphate, stick it back on, make a triphosphate form. So those are floating all around.
So just to clarify one more time. So we're attaching the 3' end of the new base to the 5' end?
No, you're attaching, so this is the new strand that you're making. So this one just got previously put in. I am putting the new nucleotide on its third carbon. Okay, so in the existing strand, that's the third carbon. I'm putting the new one on, and it's hooking in through, this is the 5th carbon here, where the phosphates are. Okay?
Oh, but then it's hydrogen bonding.
And it's hydrogen bonding here. So the hydrogen bonds occur first, right? It hooks up, make sure that's the appropriate base. If that's the appropriate base, then the diester bond forms. And so that new nucleotide has been put on the existing strand, on that existing strand's 3rd carbon. But it's hooked up to the new nucleotide's 5th carbon.
So do you always have to have at least a piece of the existing?
Exactly. Almost all polymerases have to have a free 3' end. That's hard to say. Okay, so a free 3' end in order to put the new one on. There's a couple that can do this spontaneously and can put one in there initially, and I'll show you those ones, but most of them have to have what we call a primer strand. If this is the template and this is the new strand, I have to have at least a primer base there already. Okay.
And this bond formation is from the energy from metabolism, right? So ATP?
Well, the way you've got these triphosphates is because you've metabolized and used energy. Cells are doing metabolism and have got energy around and can form it. Now, this is a TTP, right? But ATP would be what we got to put in next. Here's a T, I need an A, so I'm going to have to use an ATP. So all those ATPs had to be formed, whether it's an ATP, TTP, GTP, right? All of the nucleotides have to have been formed using energy from metabolism of other things. Okay.
And you have to have a TP, not like a D or M. Like you can just have a monophosphate.
You can't have a monophosphate. Yeah, only the triphosphate forms can be used to put in. If it's a mono, then something else, another enzyme is going to have to charge it up to the triphosphate form before you can put it in. So think about energy use. Replicating your genome, every time you put in a nucleotide, you're using up an ATP. So it's a pretty energetically expensive process to replicate your genome. One of the most expensive things your cell's going to have to do. So, okay. Now, DNA is double-stranded, And I've already told you, in order to polymerize it, we've got to get it single-stranded. So I've got to break existing hydrogen bonds in the double-stranded piece and get them into single-stranded so that I've got a template exposed and it can actually make my, replicate it. So all of what we're going to do right now is what's been worked out in bacteria. It's kind of the simplest in terms of the number of enzymes and what's going on. It's simplest to do it in bacteria first. So we'll talk about bacteria first, and then I'll tell you at the end a little bit how your cells, eukaryotic cells, are slightly different, okay? So here is what we call an origin of replication. Here's double-stranded DNA, reverse complemented. There are these regions of the genome that are called origins of replication, and they are AT-rich regions. Remember, A and T's are only making 2 hydrogen bonds, so these are going to be easier places to pull apart, less energy required to pull them apart. So they've got repetitive region, typically 9 to 13 base pair repeats of AT-rich sequence. So it'd be 9 bases of mainly A's and T's, and then it'd be repeated again, 9 bases of mainly A's and T's. And then there's also 13 base pair repeats as well. The enzyme that first opens this guy up is called, the protein is called, in bacteria, DNA-A. This is confusing, but this is a protein, and the protein's name is DNA-A. It's the guy that's going to come in and separate the 2 strands, okay? So it causes what we call denaturation. When we say DNA is denatured, we say this 2 strands are being pulled apart, okay? So it's going in and breaking hydrogen bonds, pulling the 2 strands apart. So I get local denaturation, separating the strands at these repetitive AT-rich regions. That is the first thing that comes in. He's also sometimes known as the initiator protein, or DNA-A in bacteria. That opens it up a little bit for another guy to come in called DNA helicase. And helicase is what's going to do most of the work of pulling the strands apart, okay? So DNA-A, or the initiator guy, just pulls out locally. Helicase comes in, and it's a little donut-shaped thing, and it's going to actually start what's called processing, or moving along the strand. And it's going to, one helicase is going to move in one direction, and another helicase is going to come in and move the other direction, and it's going to start pulling and breaking more and more hydrogen bonds, okay? This takes energy, so helicase, I'm not sure if it's using ATP, but it's, this is energetic too. You got to break those hydrogen bonds. So not only does it take energy to put the nucleotides in, it takes energy to separate the 2 strands. Okay. So helicase is going to unwind in both directions. Now we're going to have some partially exposed single-stranded DNA. And remember, DNA hates to be naked, okay? So you're not going to have single-stranded DNA floating around. It's always going to be coated with proteins. And so there's a bunch of different kinds, but we're just going to call the class of proteins single-stranded binding proteins. Okay, they're just going to come in, they're going to bind to the DNA, and wait until the actual copying proteins come in. They're just going to hold it, make it stable, hold it open. Because if you had helicase that just ran down the DNA and separated it and left it naked single-stranded, it's just going to hydrogen bond right back together, okay? So it's just holding mechanism to keep it apart. Okay, so I've got my exposed strand, and what we're going to do is focus in on just one of what are called the replication forks, okay? So when helicase, DNA-A and helicase start opening up, I've got this little open bubble of double-stranded, and I've got 2 sides, right? So I'm just going to focus in on one of the sides, but the exact thing is going on the other side. It's just upside down, basically, okay? So I'm going to focus in on one of the 2 forks that's going on here. Okay, so remember, DNA is reverse complemented. The 5' end is going in one direction, the 3' is going in the other, right? They're reverse. So I've got one side that's got exposed 3' ends, and one side that's exposed 5' ends. So I've already talked about helicase is the one that's unzipping this. So helicase is moving along in here, unzipping. And helicase is paired up with another enzyme called primase. And primase is one of these polymerases that can actually add nucleotides without an existing nucleotide to add it onto, okay? It doesn't have to have an exposed 3' end. It can just stick the first nucleotide in. So primase sticks RNA nucleotides in. So primase is going to sit down, and in green right here, sorry for the color, but in green right here, that is the nucleotides that primase stuck down. Okay? So it got to put that first nucleotide down without any previous one, but then it acts as a polymerase and it puts another RNA nucleotide down. Okay, so it's reading the nucleotides, putting them down, always adding the new one on the 3' end, and so it's extending. And it only does this for a short period of time, just, I forget actually how long, but it's somewhere on the order of like a dozen nucleotides. And they're RNA nucleotides. So I've got this kind of hybrid of DNA and RNA to begin with. We call that the RNA primer. This first top strand is the leading strand. Once RNA primase has put down a little primer, it actually falls off. Well, actually, it doesn't fall off, but it moves along with the helicase, because helicase is continuing to move down here. So primase temporarily just puts down a couple of RNA nucleotides, and then as helicase keeps moving down, it just goes with it, skips over a big region. Okay? So I've got a bunch of empty space. What comes in next is, and this is where the specific polymerase types come in, is DNA polymerase 3. So in bacteria, they've got several different DNA polymerases, DNA polymerases. The one that's doing most of the work here is the third one that we discovered, so it's just called polymerase 3. Polymerase 3 is going to start off where primase left off. Primase put that little RNA primer on. DNA polymerase 3 is going to come in and it's going to start adding new DNA nucleotides. Okay? Sticks those on, and up here is called the leading strand, because once polymerase 3 sits down on that primer, it's just got free reign and it can just keep putting in nucleotides, boom, 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 just pushing them down all the way down towards the 3' end of this new strand. Right? Helicase is going to continue to open it up, but it doesn't need a primer anymore. Already has a primer, it just continues to go. So we call it the leading strand. Okay. Now on the other side, do you have a question at this point? Yeah.
So in your picture here,
Yeah.
the primer is primase, the green is primase, which...
The green is the primer. Those are the actual nucleotides.
Okay. And
the enzyme that put them there is called primase.
Okay, and then the DNA polymerase 3 is the red that is now...
Those are the nucleotides that DNA polymerase 3 put down.
Okay.
Yeah. All right, the lagging strand, since it's the reverse complement, I can't immediately put down a primer here, right? Because the primer, or the polymerization has to be happening always by putting the next nucleotide on the 3rd carbon, right? So the polymerization on the lagging strand is going in the opposite manner. This is what makes it lagging, is that you can't do it in all one continuous swoop like you can do the leading strand, okay? Leading strand, same DNA polymerase 3 can just keep going as long as helicase opens up. On the lagging strand, I've got to wait for a little bit before primase can come in and stick down the primer. Okay? So primase puts down this primer. DNA polymerase 3 again comes, sits down on that primer, and starts extending it. If you think about the other side of the replication fork, this is exactly upside down. Because if we think about this guy going the opposite way, this is the leading strand if we think about going that direction. And the top strand is going to have to be the lagging strand on the other side. Okay. So the lagging strand, it can put a primer down. Primase puts a primer down. I can extend here. But then I have to wait a while for helicase to open this up. Basically, I just have to wait for room. Okay. You got to wait till the two strands get pulled far enough apart for a primase to even get access in here. Right. No access is available right now. I got to wait till helicase moves further, opens this up bigger, then primase has access to it. I can put down another primer, and then RNA polymerase 3 can come in and fill in that gap that I've had to jump over. So this is why it's lagging. It goes a little bit, and then it has to wait. Put another primer in a little bit, and then it's got to wait. So I'm doing it in pieces. These pieces are called Okazaki fragments, named after the guy who discovered them. So
So does the primase primase put down primers on both strands at the exact same time?
No, there's two primases working in here, and they're doing them at different times, right? On this strand, the primase got stuck down, and then primase doesn't have to work anymore on that side. Here it's putting it in there, and then it's got to wait, then it puts it down again. So on the two sides, they're not happening at the same time, nor is it the same primase. It's actually two different primase molecules, or enzymes.
Couldn't that primase just start a little bit earlier so that it's right
On which strand?
on the lagging strand?
On the lagging.
Like if it started just a little bit closer to the unzip?
Over here. Is that what you're saying?
Yeah.
Well, it can't because there's just not enough room in there. So we got to wait until the two strands get separated further. Gives it access, so then I've got room. It can't put it down here yet. I've got to wait. So it's got to get more room, stick it down. So, and this is part of it's energetically too. I mean, you can think of, well, why don't you just denature the two strands completely entirely, and then you wouldn't have to worry about this lagging strand problem, right? The problem is that would take a lot of single-stranded binding proteins to hold it all. It's going to want to start base pairing on itself. It's just going to get all mixed up. So we just open it in very small little replication windows here and immediately put the new strand in there so that it doesn't spend a lot of time single-stranded. Okay. Now, in a test tube, you guys are going to just denature the entire sample and separate them out. But that's in very different temperature conditions, right? If you've got DNA in a test tube, you can boil it, whatever. You're not worried about killing cells. Here, you got to be really careful about how much energy you use. You could just, overheat your cell and cook it if you tried to keep all of this single-stranded. So, yeah.
So with the Okazaki fragments, do they run into each other eventually, or is it kind of like this?
Yeah. So exactly, the Okazaki fragments. So RNA polymerase 3 is sitting down here. It's making it, and when it comes to the RNA primer, DNA polymerase 3 does not know what to do with that primer. It reaches that primer and it just falls off. It says, I don't know what to do, there's already something here. And that's why they're actually fragments, right? They're little pieces of RNA, some pieces of DNA, and then you can't hook them up because RNA polymerase 3 doesn't know how to hook those together. So it just falls off and I've got these little fragments. The primer stays there. DNA polymerase 3 falls off. The enzyme falls off. Okay. It's just when it got to here. So it sat down on the original primer, no problem, put in all these nucleotides, but when it reaches the next one, it just bumps into a roadblock and falls off. So we have to have an additional step to get rid of these. Before I move on, this is called semi-conservative replication, because in the new strands that I'm making, one of the strands is a remnant from the original strand, the other is a new one. So the DNA itself, the molecule itself, is semi-conserved. Half of it's conserved. We also call it SemiDiscontinuous because one strand is happening in fragments discontinuously, but the other half is going continuously. So half of it is discontinuously made. DNA polymerases do this very, very quickly, especially bacterial DNA polymerases. They do it on somewhere between 500 to 1,000 nucleotides per second are going on here. So very, very fast chemistry. All right, so the problem Josh brings up. What happens? So here's DNA polymerase 3, this little hand-shaped thing. This came in and sat down, filled in nucleotides. It gets here to another primer, and it's going to fall off. Can't do anything. So I have to have a different DNA polymerase to come in and resolve the problem. The pinch hitter, the substitute hitter that comes in is DNA polymerase 1. This is just the first one we found, but he's actually playing a subsidiary role. In terms of the number of nucleotides he's polymerizing, not very many. So RNA polymerase 1 swaps out for RNA polymerase 3. And RNA polymerase 1 does two things. Well, it does three things. I'm sorry, DNA. DNA. Yeah, no. DNA polymerase 1 removes the RNA primer. So it can actually chew away at those nucleotides. It's breaking the phosphodiester bonds, and it's just releasing those now RNA nucleotide monophosphates, right? So I've lost some energy there. So I'm going to have to recharge those nucleotides. So it does that, removes the RNA primers. It also then fills in the removed ones with DNA nucleotides. So it's just erasing the RNA nucleotides, filling in with DNA nucleotides. RNA polymerase 1 can fill in all the way right up to the last nucleotide, but the one thing it can't do is do that last phosphodiester bond, okay? So it's going to put in all the nucleotides right up to where DNA polymerase 3 had started. So it's going to chew away all of the RNA primer, and it's going to get right to the nucleotide, right to the next nucleotide that RNA polymerase 3 had put in, but it won't be able to make that last phosphodiester bond. It doesn't have the enzymatic activity to do that, because it would actually be polymerizing, in a reverse way, right? All it can do is add things on the 3' end. If it comes up and tries to put it on an existing one, it doesn't know how to do that. So it falls off. Polymerase 1 is done with its activity. The last guy that comes in is an enzyme called DNA ligase. And this is not a polymerase. All this guy does is come in and reforms that very last phosphodiester bond. Okay? So it's going to link up the last nucleotide that DNA polymerase 1 did and the first nucleotide that DNA polymerase 3 did. Link those together. And so it glues what we call the nicks. So a nick is where two nucleotides are there, they're base pairing, but they're just not phosphodiester bonded. So it's going to repair the nicks. Yeah, Will.
Okay. So I'm sort of going to some what you just said, so let's see if I can make sense of it. So what happens is in the lagging strand, because you have the Okazaki fragments, the primase will run into the primer and can't deal with that, so it falls off?
DNA polymerase 1 runs into the primer, yeah.
Falls off, so then DNA polymerase.
I'm sorry.
1 or 3?
I'm sorry. 3 doesn't know what to do with the primer.
Right.
So it falls off. 1 comes in, removes the primer. Yeah.
And then it connects the fragments, but doesn't finish them?
It can't connect the fragments.
It can't connect.
It fills in all the nucleotides,
Okay, that's
but then the last two have to be put together by ligase. Yeah.
Why should it do that last again? What's the bonding that would be different?
Well, one of the things is that last nucleotide, so this strand right here is the nucleotides that RNA polymerase 3 put on, right? Because the primer used to be right here. That one is not a triphosphate. It's a 5' end that only has a single phosphate on it, okay? And then also, the enzyme just does not have the capability to recognize those two nucleotides and stick them together. Now, ligase comes in and can do that chemistry. It doesn't need a 3-phosphate group on it, or maybe it adds it, I actually don't know. But somehow it's the one that can overcome that and put the two adjacent ones together. So, all right. That's a nice segue, because this is the actual crystal structure of DNA polymerase 1. And it's got 3 things that it can do. Number 1, it synthesizes DNA, right? It just polymerizes. It can make those phosphodiester bonds. If it's got a triphosphate guy, it can stick it on a free third carbon, right? It has what we call 3' to 5' exonuclease activity. Exonuclease is the activity of removing nucleotides from a polymerized strand. And when I say it's 3' to 5', it means it's backing up, right? I'm polymerizing by putting a new nucleotide on the 3' end. This says it's got exonuclease activity from 3' to 5', means it backs up. So if it recognizes that it allowed a nucleotide to inappropriately hydrogen bond, right, paired up the wrong base pair, it can recognize that. It will back up one and will actually break the phosphodiester bond that it just made, allowing that inappropriate nucleotide to leave and allow the new one to come in. Okay? So this is called proofreading activity. But technically, it's 3' to 5' exonuclease. It's just chewing away the last nucleotide. Right? There are all kinds of exonuclease activity enzymes. Exonuclease means it's removing the nucleotide, but it's removing it just from the end. Okay? Only the last one in the strand. Okay? We'll talk about endonucleases later in the semester. But an exonuclease, so it can only remove the last one. If it put an inappropriate base in there and didn't recognize it and then put another base on top of it that's buried, it can't move back to and go and remove that. That would be endonuclease activity. So it can only recognize if it made a mistake, it can edit it. So it has, short-term, or I guess only short-term memory, right? Long-term memory loss. If it immediately made a mistake, it can step back and say that was wrong. But if it made it in a couple of nucleotides ago, something else is going to have to fix that if it's wrong. Okay. The other thing is it has 5' to 3' exonuclease activity. This is what I've already told you. This is what it uses to chew away the primer, right? So polymerase 1 comes up and is chewing away the primer. It's removing the nucleotides on the 5' end. Okay, so it's 5' to 3' exonuclease. So that's removing the primer. Don't worry about Klenau fragment.
And then is that 5' to 3' exonuclease activity specific to RNA then? Like, does it stop when it's
Yeah, it recognizes and only removes the RNA. I'm not sure. It probably is only specific to RNA. Maybe under certain conditions you can get it to chew away DNA too. I'm just not sure. But its function in the cell is just to remove the primer though. Okay.
eating away at the new DNA strands too?
Yeah, you wouldn't want it to keep eating away. That would just be a waste of energy. Yeah, so it stops. It might not stop because of the RNA. Maybe there's some other recognition that it's doing, but.
So does this happen like during the entire process of the helicases, like separating out?
Well, the helicase has already kind of moved on at this point.
Or is it kind of dark
So,
separated?
but yeah, I mean, these are happening right on top of each other, right? So helicase opens up, I'm filling in and making these Okazaki fragments, and immediately RNA polymerase 1 is coming in and chewing those away too. So yeah, it's all happening together, staggered a little bit, but I mean, this is moving really quick, right? 500, 1,000 nucleotides per second. Helicase is unwinding this really quickly. RNA polymerase 3 is moving really quickly, and immediately after, I've got enough space in there, polymerase 1's coming in and resolving these. So there's not much lag time. Okay, I've only mentioned so far 2 of the bacterial DNA polymerases. There's actually 5 that we know. And I'm just going to give you an overview. I just want you to recognize that there are other ones doing other things. So we've talked about Pol1, primer removal. It's got proofreading ability. Pol3, it has polymerase activity, right? It's putting in nucleotides. It also has 3' to 5' proofreading. Okay? So if it makes a mistake by putting the inappropriate base in, it can step back 1, remove it, and put the right one in. It does not have primer removal activity though. So it can't chew away nucleotides at the 5' end. Only back up and chew them away on the 3' end. Polymerase 2 is involved in replication usually of damaged DNA. I'm not going to go into this too much, but it has both kinds of exonuclease activity because it's going to try and go in, and if DNA polymerase 3 or DNA polymerase 1 made a mistake, you can go in and hopefully it can chew away the inappropriate base, put in a new base. Okay. There's also DNA polymerase 4 and polymerase 5. These are typically turned on when you have damage of DNA that you can't resolve. You can't just like clip out the nucleotide and put a new one in. If you've got really bad damage and you still need to replicate your genome, it's involved in kind of like bypassing the damaged parts and kind of salvaging what's left. So this is also, both of them are specific to damaged DNA. Okay. trying to resolve or at least skip over. So those are the 5 that are known. Just going to talk real quick about replicating bacterial genomes, bacterial chromosomes. Bacteria chromosomes usually only need one origin of replication. You only have to have one place to separate the genome because they're usually small and circular genomes, okay? So you've got one origin of replication, you open it up, you've got a leading and lagging strand going one way, a leading and lagging strand going the other way, and so you're replicating around. Now eventually those are going to move along and they'll meet up with each other. The 2 original template strands will have completely denatured from each other, and the polymerases will fill in, and you'll make 2 completely new circular genomes. Okay? This is called bidirectional replication. This is when the genome stays circular through the replication process. So it always stays as a circle. Both template strands stay as a circle. You just denature them, make 2 new. Don't worry about gyrase and topoisomerase. We'll talk about those later, actually. There's another way that bacteria can replicate their genome. They can do it by kind of making one of their strands temporarily linear. So you can actually clip, so here's a circular genome. You could actually clip off and make a nick between the 2 strands, and you could unravel one of those template strands out, and it's no longer circular anymore, right? So if I've imposed a nick on one of the 2 strands, when it denatures, one of them is going to be single-stranded. The other template strand stays double-stranded. This allows you to replicate things quicker, but this is called rolling circle replication. So one of them, as you replicate it, stays circular, the other one's linear, and you can just make multiple copies. You don't have to wait to open up another origin of replication, basically, right? If I do it the bidirectional way, I've got to have an origin of replication that opens up. Then I've got 2 forks that I can go around, okay? But then I've got 2 complete circles. I have to open those up now at the new origin of replications. If you do rolling circle, you've already created a nick. You can just, the circle, you can just keep rolling around it, right? The circular strand is still circular, but I don't have to pull anything apart anymore. I just keep going right around, round and around and around. And then the linear strand, you fold back on itself. We won't go into it too much, but I just want you to see there's 2 ways to do it. Concatamer, yeah, that's, you put a bunch of the enzymes together, they stick together, and they're doing both strands at the same time. In a way, it like eliminates the need for a lagging strand, because you've just opened both of them up. So.
That replication doesn't need an origin replication?
Well, I mean, you still have to pull the other strand off, but basically you don't have to start over, right? Here you start with a circular genome, you end with 2 circular genomes, and then everything has to reset. You got to start it over. In the rolling replication, this guy just keeps moving around and around and around and around, and you never have to open it back up again. Okay? Now you still have to denature and pull them apart, but when bacteria are in their exponential growth phase, the bacteria are just replicating their genome as fast as they can. So you'll just replicate off a genome, and then the cell will divide, and then it's still replicating its genome. It never stops, right? So as soon as the genome's replicated, the cell divides, and then those 2 start replicating their genome. And as soon as the genome's replicated, they divide. They're using this really rapid rolling circle. It's just the limitation of growth is just how quickly can you get your genome made. If there's lots of nutrients around, they're just going like crazy. Contrast that with eukaryotic replication. So this is cells like yours, yeast. Eukaryotic organisms have a much bigger genome typically. It's much more compact and condensed. Cells grow slower. And so they have checkpoints. And I'm not going to talk about the cell cycle too much, but I just want you to know that eukaryotic replication is very tightly regulated. There's none of this like just replicate your genome as fast as you can and just keep going. Eukaryotic cells are Typically much slower than that in terms of like, wait till the whole cell replication process is done, wait till the cell divides, then start over and replicate your genome again. I'm not sure exactly what yeast do. Yeast can go pretty quick in their eukaryotic. But in general, eukaryotic genomes replicate slower and wait for the cell cycle to complete. Okay. Yeah, Will?
Is there an advantage or disadvantage to replicating quickly? I would imagine I can see an advantage in replicating quickly, but you know how you said you wait and it's a slower process. Why?
Yeah.
Is that the case?
Bacteria, well, I can kind of speculate on the reason why. Bacteria don't really care too much about the fidelity of their genome. If they make a mistake here and there, that basically, they made a mistake in replicating their genome, and the daughter cell that buds off of that has got a mutation. And I'm anthropomorphizing a little bit, but who cares? That's not me anymore. That's another individual, right? So if mistakes are made, it's not that big a deal. So DNA polymerases go much faster, cell cycles much higher, and so mutation rates are higher as well. And if a mutation rate is lethal and a bacteria dies, it's not really the end of the world. There's lots of other ones that are still going, right? In a eukaryotic cell, and usually eukaryotic cells are in organisms that are multicellular, you're keeping all of your cells around, right? So if you made a mistake in one of the genomes of your cells, that mistake is going to stick with you, right? Either the cell is going to die and it's not going to be able to do the function you wanted it to do, or you've made a mistake in a gene that's going to cause cancer or something, and now you've got a toxic cell in your body. So eukaryotic slow down. There's lots more proofreading, making sure the genome was copied correctly before you do anything else. So that's kind of anthropomorphized in a way, but especially for a multicellular animal, that's really important.
Seems like there's a lot less mutation.
There's a lower mutation rate. Some of it's because the polymerases are better and they don't make mistakes as often, but also because there's other enzymes that come in and proofread again and say, okay, before we end this cell cycle and start again, let's make sure the genome was copied appropriately at the last cell division. So that's what I mean by tightly regulated. Right.
Is that why bacterial resistance to antibiotics can grow so fast? Because there's lots of opportunities.
Yeah, so the question is, does that mean that's why bacteria can adapt to antibiotic resistance? That part of the reason is that, yeah. The other reason is sometimes they're sharing information between each other. So a bacteria who survived an antibiotic environment has gotten a resistance gene that can often be conveyed to an adjacent bacteria, and so you share the resistance. That's a different mechanism, but allowing it to adapt to a new one that it's never seen before, yeah, part of it's the higher mutation rate. All right, in eukaryotes, expanding on this theme of more and more complex, eukaryotes have at least 15 DNA polymerases. I haven't actually checked in a couple of years. We may have found more than this. They have pretty good bacterial counterparts for most of them. So polymerase A basically is the eukaryotic equivalent of primase. Okay? It can put in the RNA primers. It also has DNA polymerase activity as well, so it synthesizes its own primer and then switches and becomes a DNA polymerase. So it's a little bit better. Around 20 nucleotides, it drops off though, because it really is just to lay down a primer. And then the eukaryotic equivalent of polymerase 3 comes in, and in eukaryotes, this is delta, polymerase delta, or polymerase epsilon. So polymerase delta is basically the equivalent of polymerase 3. It's highly processive, does most of the work. It's got proofreading activity. Epsilon, it seems like maybe sometimes substitutes in the lagging strand. And I'm not sure if it has primer removal or not. And then you've got all kinds of other ones. What is that? Alpha, beta, gamma is mitochondrial DNA. Beta is in DNA damage. Then you've got nu, iota, kappa, rev1, zeta. You gotta know Greek to know this. What is that? Sigma, lambda. Is that psi? Phi? What are these? sigma, that's sigma. What's that? Lambda, sigma, mu. So we got at least 15. There's others in the genome that look like maybe they actually are DNA polymerases, but we're kind of not sure whether they get expressed or not, because these are very rarely expressed, right? Most of the time it's the big players. It's alpha, it's delta, it's epsilon. And some of these other ones, we don't even know when they get turned on. They seem to only get turned on in response to certain kinds of damage or at certain points in the cell's life. So we're still really trying to figure out what all these do and how many there actually are. And it kind of varies between organism to organism too. So, but at least 15 distinct ones are known at last check. So, yeah.
How intense the gene is to know this slide?
I want you to know the main eukaryotic equivalents to the bacterias. Okay? So I want you to know how bacteria do it, polymerase 3, polymerase 1, primase, and then just know what the names of them are, the equivalents are in eukaryotes. They're up there. Polymerase alpha is the primase. Delta is the polymerase 2, basically. And epsilon is kind of the equivalent of polymerase 1. Yeah, I just want you to appreciate how much more complex eukaryotes are. We could spend a whole semester just going on, how complex eukaryotic expression is. So, all right, last slide, and we'll get to PCR and telomerase next time. In eukaryotes, they don't have a circular genome. They've got multiple and very long linear chromosomes. So instead of having a single origin of replication, to do this in a timely manner, you don't want to have to wait all the time for a single polymerase to do the entire chromosome, right? So eukaryotics are going to have multiple origins of replication. These are still AT-rich regions. I'm not sure if they correspond to the same 9 base pair repeats and 13 base pair repeats, but they're AT-rich repetitive regions that get opened up. And so I can have 2 templates. I've got 2 replication forks here. And then adjacent to that, another initiator protein is going to go in there. The eukaryotic equivalent of DNA A is going to go in there, separate those 2, start opening up a new replication fork. And those eventually are going to meet up and make a big one. So I'm going to have multiple locations along the entire chromosome to start replicating. So I'm doing this all at the same time. All right, we'll talk about how you replicate the telomeres, the very ends, next time.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.